Hi, I'm Dr. Jack West, medical oncologist at City of Hope Comprehensive Cancer Center in Los Angeles. This is West Wind, a podcast focusing on the intersection of cancer care, emerging technology issues in medicine, and healthcare policy. You can find information about subscribing at beaconmedic.com or wherever you find your podcast content. Please leave a review, especially if you like this, and we'd also love to get your feedback at westwindpodcast@gmail.com. I'm happy to be here now with Dr. David Gandara, who is Professor of Medicine Emeritus and a Senior Advisor to the Director of Medical Oncology at the University of California, Davis, in Davis, California. He is also past president of the International Association for the Study of Lung Cancer, ISLC, and for many years was the chair of the Lung Cancer Committee for the Southwest Oncology Group. Very few people have had the kind of influence that you have had on the field of thoracic oncology. So thanks for taking the time.
My pleasure, Jack.
Let's start with the issue of developing the careers of so many other people in the field. You've done that directly for me through the Southwest Oncology Group. You've participated heavily in and been on the board of directors for ASCO, and of course as a past president of the ISLC. So, and at your own institution, you have shaped the trajectory of many prominent people in the field. Can you comment on the importance of mentorship, maybe that you've received, but certainly that you have exerted the influence on so many people over the last few decades?
Well, mentorship has been important to me throughout my career, and I hate to say this, but it's partly because I didn't have strong mentors, at least in thoracic malignancies. And I feel like to a certain extent I had to use the term, pull yourself up by your bootstraps. Because of that, when I reached the level in my career where I could actually offer mentoring to others, I felt like it was something that I had to do. And I have been blessed over the years with having fantastic people to mentor, both within my institution, outside my institution. As a matter of fact, right now, I have 3 people outside my institution who have awards for various projects, and I'm their mentor. And also, I believe it or not, I'm actually mentoring a surgical oncologist now in GI oncology, because to tell the truth, it's like reviewing a grant. You don't have to be an expert in that area. You need to know how to mentor. So anyway, I think it is really important. We're here at the ISLC Targeted Therapies meeting in Santa Monica. And I can tell you 3 or 4 times already in this meeting, people have come up to me. Maybe it's because I'm looking older now. And they've said, I just want to let you know, you are the first one who invited me to such and such. You got up and defended me when other people attacked my paper that was presented, etc. So that's very gratifying. And I try to teach mentoring skills to my colleagues. To a certain extent, people's personality lends themselves toward being mentors or not, but everyone can do it. And the key is to put yourself in the position of whoever the mentee is and figure how can you help them. Sometimes it's making a connection. Sometimes it's a very formal process by which they have a grant that's a mentored grant. ISLC has been, I think, particularly good in having a series of awards that are linking junior investigators to mentors in many different ways. And I've been happy to participate in that process.
It just seems that in addition to the way to create a legacy by creating data that influence standards of care, by perpetuating the field and the development of future investigators, it's another huge way to build a legacy. Let's turn to the issue of running clinical trials. And you presided over the lung cancer committee for the Southwest Oncology Group for many years and are still very influential in it. The whole enterprise of running clinical research trials has changed a lot in that time. It used to be largely standard drug, standard chemotherapy plus or minus drug X for looking for a 6 or 8 week improvement. And now we have narrower and narrower populations to study. And it's the regulatory challenges and the costs are also a challenge. Is this still feasible for community-based oncologists to do in terms of the support staff, the costs, and the populations when it gets narrower and narrower? And maybe you could speak to the evolving ways the trials are done, like LungMap too.
The clinical trial enterprise has never been more complicated than it is now. I'm sure you saw the recent publication that said for checkpoint inhibitors, there are currently ongoing 2,250 trials requiring a total patient resource of 400,000 patients. 750 of these trials are in lung cancer. Do all these trials need to be conducted? Will they help us take better care of our patients? I would submit to you the answer is no. Now, whether we only need 1,000 versus 2,250 or how that sorts out, I can't say. But although I have worked with industry closely in many projects, the primary avenue for clinical research for me has been the National Cancer Institute cooperative groups. We can't even use that term anymore. They got away with cooperative. But now it's under the umbrella of the NCTN, which blends all the groups together. And in order to be successful in clinical trial research in that sort of environment, we have to be more clever. We can't do trials that industry can easily do. We have to pick patient populations that are less applicable to industry trials. We have to engage more in precision medicine approaches. We have to find out how everything that we propose will be complementary to what is ongoing with a growing industry in oncology. And I'll just give you one example of this, and that is the ECOG-SWOG joint trial, first-line therapy of immunotherapy and immunotherapy combinations. It's about to be activated in a month, called INSIGNA. When ECOG and SWOG went separately to the Thoracic Malignancy Steering Committee, I think you were probably on it, Jack, when this happened.
That's right.
We were told, well, you have two competing proposals here. Number one, we want you to work together. Number two, we want the endpoint to be overall survival, because that's what benefits patients the most. And number three, we want you to devise a trial so that it remains important, regardless of what all the industry studies show. And believe it or not, I think we did it. It's going to launch next month. I don't want to go through the details here. But what I'm saying is that you have to be agile. You have to be able to adapt. And here I'll give you a second example. That is the public-private partnership, which is LungMap. So this blends in partnership from the National Cancer Institute with the foundation of the NIH, who's the purse holder for funds, pharmaceutical industry, and biomarker companies, and the FDA being a close consultant. Friends of Cancer Research, patient advocacy group, also a leader in this joint collaboration. In that project, we have used the opportunity of precision medicine to do a series of sub-studies under an umbrella master protocol mechanism. That's the sort of thing that is very difficult for industry to reproduce, because, as Roy Herbst likes to say, we have a funnel for patients to get genomic testing. And if this funnel can pour in tumor tissue or blood from 2,000 patients a year, we will find common abnormalities, we will find rare abnormalities in sufficient numbers that we can do trials in those populations. And no matter how good an industry mechanism could be, and because they do have their own basket trials and so forth, they don't have 450 active sites around the entire United States, the majority community-based, that have a protocol that is ongoing now, one protocol, umbrella protocol, for 4 years. And that every time there's a new sub-study, the central IRB, since it's approving, within 3 weeks, it can be reactivated in all the sites. So that's the sort of thing, and I just use it as one example, where we have to be complementary to what industry does. On the other hand, I don't mean to divorce industry. You and I are both consultants. Mm-hmm. How else we can advance the care and hopefully future cure of patients is to give our input into industry and help them develop the best trials that they can do.
Well, I would imagine also that having these kind of umbrella protocols like LungMap lowers the labor intensity of community centers to be able to participate. It allows them to get access to treatments for rare mutations that you otherwise couldn't hope to have an individual trial for a 1% population. But if you participate in LungMap, you can get treatments for your patients, participate in clinical trials without having to have everybody travel for that.
I think that's right. I will just say that one aspect of LungMap, which I'm very happy about, is we realized early on that we would never have enough sub-studies ongoing, because they open and close all the time, of course, to account for every patient. But we wanted every patient to get something. So we have a series of sub-studies that are primarily related to immunotherapy, but not necessarily all, where they are so-called non-matched studies. So what that means is, and you may have a biomarker embedded, but it's not required for the patient to go on the trial. So what that means is, if Mrs. Jones comes in and she gets next generation sequencing done, and she doesn't have the requisite biomarker that would put her into a genomic-driven sub-study, she would flip over to one of the immunotherapy studies, so that as long as we continue to generate new sub-studies on a regular basis, it allows us to be very attractive to patients because they know they will get something, and also to practicing oncologists.
That's great. Let's turn to blood-based testing. We're in a new personalized medicine genomics era, and for everybody, limited tissue is a major challenge. That's probably especially true the further you get from the tertiary care centers. So what would you say now is the role and the strengths of blood-based testing? What would you say are still the limitations in its use?
Well, blood-based testing, or so-called liquid biopsies, which could today in 2019 be used both for detecting molecular abnormalities and perhaps immunophenotyping. The strengths are that if there is inadequate tissue, you've established a diagnosis of lung cancer, let's say adenocarcinoma, but there's inadequate tissue for broad-based molecular testing, which is the standard of care today, that you could use blood-based testing as a substitute in some cases, and we can go through those maybe in a moment. The second is that we know lung cancer in particular has tumor heterogeneity, so that not every cell or not every site of metastasis carries the same oncogene, and blood-based testing gives you a global perspective. So it helps to aggregate that. We also know that blood-based testing, because it's easily acceptable and low risk, can be used if there is justification for monitoring. Now, this is still an area that is evolving, but we know from study after study, and this is now true also, it's emerging for checkpoint immunotherapy, that if a patient enters a therapy and they have, let's say, an oncogene that's in their cancer and it's in their blood, and if at 2 months that oncogene is still there quantitatively, the variant allele frequency is either the same or it's gone up or it's not eliminated, they're going to do poorly. Now we know the same thing is true for checkpoint immunotherapy. Regardless of what the mutational components are, that circulating tumor DNA should go down. And there's even some data that tumor mutational burden should go down. Right now, you might say, well, what if I have a patient with an EGFR mutated lung cancer, and let's say they're on a first or second generation, and I detect T790M in the blood, but the patient's still clinically doing well. Right now, it wouldn't be standard of care to change them to a third generation like osimertinib. But in the future, it might be, because there are trials, I call this biomarker switch therapy, trying to see if early switch, early intervention to an agent to address that problem, could be MET amplification, if that actually would, at the end of the day, improve survival. So blood can do that. You know, you can only rebiopsy the patient's cancer so many times.
Let me just ask, in the community, oncologists are being reached out to, marketed from various potential sources for this testing. Is this platform of plasma testing something that that a community oncologist or any oncologist should feel comfortable getting from a CLIA-certified lab, or is this really something that we should feel wary about from anybody who hasn't been publishing in this space at this point? Is this something where you would envision this being a winner-take-all or a really clear gold standard, or is it very variable and any of these is going to give you similar results?
Of course, there's no easy answer to that because it's a rapidly evolving field. The sensitivities of the blood-based assays are dramatically better now than they were even 18 months or 2 years ago. So that there are essentially no false positives for driver oncogenes. And the sensitivity is such that even very small amounts of these driver mutations in blood are actionable. And there are wonderful data now for the oncogenes to show that it's meaningful. In other words, that patients respond to ALK inhibitors or EGFR inhibitors by finding these abnormalities in the blood. Which test you use and where you use it requires education. So Dr. West, who is interviewing me, and myself and 2 of our colleagues are running an ASCO educational session at ASCO 2019, specifically on molecular testing and the ins and outs of various technologies and blood versus tissue, and Dr. West is sorting it out at the end about what do I do with this information. So I think it's like, it's no different for an oncologist to have people from industry coming in for new drugs.
Right.
And so you evaluate it, you look at the literature, you make a clinical decision. I'm going to use drug X versus drug Y if they're both available for that treatment situation. And I think it is true for these tests as well. You want a test that you can interpret on your own, and you know how to use it, but you also want a resource so that if you don't know about this variant of BRCA, there is an expert that you can call and say, what does this mean to me? And it would be the same as you have an unusual adverse event on a patient with drug, and you call the company and you say, have you ever seen this, and what does it mean?
But there are so many variables now. I mean, molecular oncology has added a dimension that I think education and being able to tap into some of that collective wisdom on demand, whether that's a database like OncoKB or various others, My Cancer Genome, or a molecular tumor board. It's going to be really important to diffuse that collective information out to the rank and file community, because it's a bewildering amount of information.
Right, and I want to clarify too that I am speaking about lung cancer, and specifically non-small cell lung cancer. There's been a lot of negative press and publications recently about the flop of precision medicine. So, precision medicine can only be applied effectively if there's something to do. So you can write all the articles you want about the lack of effect of precision medicine in prostate cancer or even carcinoma of unknown primary, because you're not going to have enough hits to justify doing testing. Well, in lung cancer, you do have enough hits. And I think going back to the days where you say, I'm going to do a KRAS mutation test, and if it's positive, I'm not going to test any further, those are long gone.
Yeah. Well, I agree. I think we're living through history right now, and I think we're moving into an era where NGS is about to become routine and a gold standard, at least for lung cancer and increasingly in other settings too. But we're going to have to struggle to keep up and to educate everyone about it. So thanks so much for taking the time. It's been great.
My pleasure.
Thanks so much for joining me today for this West Wind podcast. We hope you'll be interested enough to subscribe either by checking out the options at beaconmedic.com or looking for West Wind Podcast on iTunes or wherever you find your audio content. Please take the time to submit a review, and we'd also welcome your feedback at westwindpodcast@gmail.com. Until next time, this is Dr. Jack West. Thanks for listening.